Welcome to Spring LIB.
Okay, so problem 80 is a bit of a long one. It has several parts to it here, so let's dive on in. And so the main thing happening here is that we have this cyanobacteria, right? And so here we have these two cyanobacteria cells, and these two active cells sometimes will form a heterocyst between them to aid in creating ammonia and processing the nitrogen in nitrogen-limiting environments. Okay, so that's sort of succinctly what's going on here. And then this one is asking us for part A to sort of define the four modifications happening by the vegetative cells, so that's these active cells, to the heterocyst. So like what's happening to the heterocyst? And of course, they have these numbers here, 1, 2, 3, 4 at the bottom. Okay, I'm just going to write them off to the side though for space. And so the first thing we can see here is that while the active cell brings in the carbon dioxide, it's only putting carbon into the heterocyst, right? And so it's doing two things here. First of all, it's putting carbon into the heterocyst, okay? But then it's also omitting the oxygen, okay? And then the third and fourth feature can be seen at the bottom. And so it's putting both the hydrogen and nitrogen here into the heterocyst, right? So both the hydrogen and nitrogen. Started to write that a little funny. Okay, so hydrogen and nitrogen. And so that makes sense because we need those to make ammonia, right? So those are elements that are going in. And then part of A wants us to sort of draw arrows to where this is happening, right? So we draw an arrow from 1 to where carbon is going in. That's going to be here. We draw an arrow from 2 to where the oxygen is being omitted. So that's sort of in this process. 3 and 4 is sort of happening here, right? Okay, I'm just going to remove those arrows now, just due to clutter, but that's where they would be. And then part B wants us to describe the role of each of these 4 steps, okay? So the role of carbon, and this is really just wants us to describe it in one of two ways. Like we're just choosing categories of whether it regulates oxygen or synthesizes ammonia, okay? And so the role of carbon here going in, you can see that carbon plays the role in glycolysis, right? Which of course would be the need for our electron transport chain to get energy, and that energy then will synthesize our ammonia, right? So this one actually plays a role in the ammonia synthesis. Okay, now omitting oxygen, the inside of the heterocyst is actually anoxic, so it's an oxygen-free environment. So by omitting it, we're maintaining that. And so this is in oxygen regulation. Okay, oxygen regulation. And then it should be of not much surprise that then the hydrogen and nitrogen are part of the ammonia synthesis, right? And that's because ammonia, of course, is made of hydrogen and nitrogen, okay? And that can be actually seen here. All right, so that's parts A and B together. And then C is a little bit different, so we have a different graph for that one. And this gives us a little representation, like an incomplete graph of this cycle that occurs, okay? So this is the Krebs cycle, and it wants us to fill in these elements at the top here. Okay, so these elements with these 4 features, and it gives us a list. It's almost like a logic puzzle, okay? So we are told that het R and pet S are transcribed when the 2-oxoglutarate concentrations are low, and we can see that het R and pet S are currently not being transcribed, okay, because there's nothing there. So that sort of tells us that the 2 oxoglutarate concentrations are not low. Okay. So that tells us that nitrogen must be limiting. Okay, they're increasing, which is based on the first statement. And so then we can see that on our fourth statement, nitrogenase, which is being expressed here, okay, that's transcribed when het R concentrations are high. Okay, so this is happening when het R is high, and when pat S is low. Okay, and then we're also told then in the last statement that pat S concentrations are high when the NIFX genes are not transcribed, but the NIFX genes are being transcribed because we have the nitrogenase. So it really is repeating part of the previous statement that tells us that the pat S is low. And you might be thinking, well, what about pat A? That never gets mentioned. And so the pat A is actually not a transcription factor. It's a metabolite, right? Which is usually a small organism that sort of aids in metabolic features, a very small thing. Oh, so then we're just sort of trying to figure out which of these 4 items on the left fill in each of these 4 kinds of boxes here. Okay. And so since the metabolites are quite small, we're going to put pat A here. Okay, and then we know that we have an increase of het R, a decrease of pat S, and that the 2-oxoglutarate concentrations are not low, right? So the thing we should have the least of is going to be pat S, right? So it's going to be probably this one here that has sort of a chunk taken out of it, right? It has a less than regular concentration. If you wanted to think of like a normal puzzle piece, this would be a regular amount, okay? And so we have a chunk taken out of it, that means it's low. So that means this one is going to be our pat S. Okay. And then when we have this puzzle piece, but instead of having a chunk taken out of it, we sort of have a globule on top. So that means it's like increasing, right? There's more than regular. Okay, so that makes this one, that's going to be our het R, because that one is increasing. And then these are going to be our 2 oxoglutarates, right? Because that concentration is increasing in number, right? That one's increasing. Okay.